number 14. We're going to talk about acute inflammation, the causes, mechanisms, vascular changes, cellular events, neutrophils, and everything else. So I want to go over what is inflammation. And basically what it boils down to is if you imagine that you have some tissue right here, this is your tissue, and within that tissue you have a little bit of blood supply, and you have stuff in your blood. So now we're going to imagine that our tissue gets damaged. Got a little damage on my tissue here, and now I want to take care of that damage. And I've got all the stuff to take care of that damage sitting right inside of my blood supply. It's in here. So I have what do I have in here? Let's zoom in. Let's take a microscopic look and zoom in. What do I have in my blood supply? I have proteins. So for example, I have proteins that include complement. I have the fluid, H2O. I don't know why that's so important, but I guess it is. And I have neutrophils and other leukocytes. And of course, it's you never want to forget that your blood also carries energy. So it's carrying oxygen and nutrients, and so you're getting a lot of energy from the blood too. You want to get all of that stuff. If we look, we want to get all of that stuff out of there. We want to get it over here. And inflammation is what we call that process of taking important repairing things from the blood and putting them in the tissue. That's inflammation. This is Elmo, used without permission. So why Elmo? No, why inflammation? Why is basically to fight invaders and initiate healing. So the causes of inflammation would be trauma, tissue damage, or infection. So why we have inflammation? To take care of those things. Now I'm just going to go ahead straight up and tell you I do not have enough time in 10 to 20 minutes to go through all of inflammation. I'm going to go as fast as I can, but I recommend going in, doing chapter 2 of Robbins, and more than that, go through and watch the 3 videos that Sitar has on pathoma. It will be awesome. But the thing that Sitar points out is high yield is what causes inflammation. The 2 big causes, necrosis and infection. Of course, any type of tissue damage, whether it's a burn, an injury from the cold, or any type of, you got hit in the head, anything like that can cause tissue damage and cause inflammation, acute inflammation. But specifically you're going to be tested on is necrosis and infection for boards. As far as clinical manifestations, there's something called the cardinal signs. These are like noted in Egyptian papyrus like 18 million years ago or something, whenever apes could first write and that kind of thing. But the cardinal signs are rubor, tumor, calor, dolor, and functiolasa, which is redness, swelling, heat, pain, and loss of function. The heat can mean the heat that's local, or it can also mean heat from a fever, depending on how systemic the infection gets, or not the infection, but the inflammation gets. And now that I made that slip of the tongue, I need to go ahead and point out, inflammation is not the same thing as infection. Infection can cause inflammation, but like I just said, so many other things. This is all from fish oil, or inflammation, I don't know. Used without permission. Now last week I did a presentation on the exudative versus transudative whenever you're talking about fluid leakage out of the vasculature and into the tissue. And in inflammation, if you just think about it, think it through, you're going to realize that it's exudative. If you remember right, anything that tends to be local or have an effect on the endothelial barrier will cause exudative. And so what we're going to have is exudative. Now, what's going to be the difference between acute and chronic? And I like Sitar makes it really, really simple. He points out that in acute, so if you got your blood vessel here, let me make my blood vessel a little bit thicker. You got your blood vessel wall here, and inside of that blood vessel you've got neutrophils, or you have lymphocytes. Now, if you have neutrophils coming into the tissue, it's acute. If you have lymphocytes coming into the tissue, it's chronic. Those are your big distinguishing features. That's like your big idea. So there's 3 brownie points for the first person that knows what you're looking at here. I'll give you 2 more seconds. 1 more second, and this is, you're right, polymyositis. Good job, whoever said that. Now what I want to point out here is all of the neutrophils that are in this thing. This looks like an acute inflammation. Also, what else comes out of the vasculature with inflammation? You get neutrophil infiltrate, and you get edema, and you can look and see all of this kind of like empty space. This is basically going to be fluid filling up around the muscles, and this is a polymyositis. So we got basically inflammation of the muscles. Now let's see who was really paying attention. What did I just say that was wrong? First of all, I said that this was neutrophil infiltration, right? Polymyositis, it is mediated by CD8 positive lymphocytes. So this would be a chronic inflammation. So there's trick question of the day. And I mean, it wasn't much of a trick question. I mean, we just studied this like, what, a year ago. So you should know this, right? Okay, so we're talking about the characteristics of acute inflammation. I already told you it's going to be neutrophils. It's going to be also have the cardinal signs of inflammation, especially edema. So if you have neutrophils and edema, you're looking at acute inflammation. If you have lymphocytes and edema, you're looking at chronic inflammation. So let's start with an injury of some sort and work our way up to the cardinal signs. And I think that's as far as I'll be able to get in this time period. All right, histology question of the day. What cell am I looking at? Clue, it's covered in IgE and it's filled with granules. That's right, it's a mast cell. Now what can activate a mast cell? So first thing would be tissue trauma. So tissue trauma of any kind can activate a mast cell. The second thing would be C3A and C5A. So that's supposed to be a C5A, C3A. So complement. And then also the cross-linking of IgG. So if you cross-link, and if you remember the IgE, so let me make this a little bit clearer. You have these IgE, and if you have one over here, and if some type of infection or some type of allergen can come here and cross and link to this one and to this one at the same time, then it will degranulate. And what will it do whenever it's activated? Whenever you get the C3A, C5A, the trauma or the cross-linking, it will degranulate, releasing, that's right, histamine. So here's a pretty picture. I tried to find where it looked like histamine was being released, but I think it just looks like a cell is vomiting. Now histamine's going to do two things, right? The first thing it's going to do whenever you get the histamine release, it's going to cause vasodilation at the precapillary arterioles, so at the arterioles. And it's also going to cause vascular permeability. So permeable, we'll do that, and that's going to be at the venous side, so at the postcapillary venules. So let's look and take a really close microscopic look at exactly how this is all happening. So first of all, at the arterioles, so we have the arterial supply right here. We have these cells in here, and I'm going to go ahead and get my microscope out and zoom into the cell. And I will remember that I have this receptor on my endothelial cells for it's an H1 receptor, and it will receive histamine. And if I draw this a little bit better, you'll remember that the H1 receptor is a 7-transmembrane receptor, which is also known as a G-coupled protein receptor. Specifically, it's a GQ coupled protein receptor. You remember the GQ protein receptors, GQ, you just want to remember all of the C's. For example, phospholipase C, calcium, and protein kinase C. So it's going to go through all that pathway to activate protein kinase C, but it's also going to release a lot of calcium into the cell. Now this particular image I am reusing with permission because I took it from the Nature Publishing Group website, and I'm pretty sure that they would probably sue me. So I applied for the permission. And anyhow, you'll see that calcium really important with this thing called eNOS. This is endothelial nitric oxide synthase. So on the arterial side, histamine through the H1 receptor, GQ coupled protein receptor, it is allowing calcium to enter the cell, and it's activating eNOS, which eNOS will produce nitric oxide from arginine. So it takes arginine, turns it into nitric oxide. So just remember, arginine gets converted into nitric oxide from eNOS. And there's actually 3 different types of nitric oxide synthases. I just kicked something. There are 3 different types of nitric oxide synthases. There's an iNOS, an eNOS, and an nNOS. But eNOS is what we're really worried about here. And then you remember back from block 1, block 2, what happens? NO, it goes in and it stimulates something called guanylyl cyclase. Guanylyl cyclase, I'm not going to write it all out. Cyclase, I can't even spell. And then guanylyl cyclase, you'll remember there's this thing called GTP, and it gets converted into cyclic GMP, right? And cyclic GMP causes vasodilation, or it causes smooth muscle relaxation, right? Smooth muscle relaxation, that's kind of the idea behind Viagra, is what happens with Viagra is this cyclic GMP, it gets converted into GMP. It gets converted into GMP by phosphodiesterase. So let me make some more room here. So phosphodiesterase does this. And you remember the principle behind Viagra and such was that you could inhibit phosphodiesterase and build up your cyclic GMP instead of breaking it down into GMP. So the point is, the ultimate point that I want you to remember is that histamine on the precapillary arterioles will cause vasodilation through relaxation of the smooth muscles, and that occurs through eNOS-induced nitric oxide production. When I told you I applied for the license to reuse that figure, yes I did. That thing took 15 minutes of my life, and here it is. Like, you can't even see the whole thing, but here it is. And for whatever reason, I forgot to put it on the next slide, was also that you got histamine causing endothelial leakage on the venules. And that happens probably through two mechanisms. One is the cytoskeletal rearrangement. You'll remember that F-actin is the filamentous actin that is all stretched out and like making structures and stuff, and then G-actin is the globular actin subunits that when it gets broken down, and so you can have different signals causing breakdown and repolymerization and cause the shapes to change and everything else. So that's one mechanism. And the other is that there was some evidence that tight junctions are being broken and stuff like that through all of these signaling cascades as well. Specifically, histamine was the one I looked up just to make sure. I just wanted to like, how is this working? Because the books don't really tell you how, it just tells you what, and so that kind of irks me. Okay, so but how else do we get vasodilation? So we had our mast cells. Our mast cells can release histamine, but also just tissue damage in general. What does it cause? Calcium. And what does calcium do whenever it comes into the cell? So you damage your endoplasmic reticulum a little bit, you let out a little bit of calcium, you got some tissue damage going on. What is that calcium going to do? What's it going to activate? If you said phospholipase C, you are right, and I think like the whole mechanism biochemically involves phospholipase C, but what I'm looking for is phospholipase A2. Phospholipase A2, why? Because phospholipase A2 is going to cleave some membrane stuff and it's going to release arachidonic acid. Now I pointed out that this is dead and dying tissue, but you know also mast cells have two jobs. They have a first job and they have a second job. Their first job is histamine release, and then after that histamine release is done through complex signaling, they have a delayed mechanism where they also start acting and making arachidonic acid through a phospholipase dependent mechanism. So for right now, what I want you to remember is that tissue damage and mast cells. And remember, it's the mast cells, like the secondary, the delayed, we'll put delayed mast cells. These two things produce arachidonic acid. So I don't even have to start off with mast cells, but I probably will because tissue damage will activate mast cells to degranulate and release histamine. But let's say there were no mast cells around, just tissue damage in general. We're going to have calcium flood in the cell. It's going to activate phospholipase A2 and probably going to be in a mast cell. But regardless of where it's at, phospholipase A2 is going to cleave arachidonic acid, and arachidonic acid then will be able to go down one of two different pathways. Arachidonic acid can either be acted on by cyclooxygenase or lipooxygenase, specifically 5-lipooxygenase. And you'll remember that there are several cyclooxygenases. There's cyclooxygenase A1, A2, B5, 9, 10, 11, 12. And like aspirin inhibits cyclooxygenase 2 irreversibly. And if you go the cyclooxygenase pathway, you can get prostaglandins. So prostaglandins, and there's three important types of prostaglandins. There's a lot of types, but there's the D2, the E2, and the I2. So I put these in alphabetical order, but if you rearrange these, you get basically that you're going to die. D-I-E. So die, and remember that the last letter on there, that's the severe one, because before that you just have D. But once you put that E on there, it's die. And that's an important one because prostaglandin E2, the E2 right here, that causes pain and fever. Now all three of these cause vasodilation and vascular permeability. Again, vasodilation at the arterial, permeability at the venule. But you'll remember that you die once you throw that E on there. The prostaglandin E2 causes pain and fever. Now what if we go down the lipooxygenase pathway? Let's kind of bring this over here. The lipooxygenase pathway, you can get leukotrienes. So leukotrienes, LT, leukotrienes, you can get subsets of those. So you can get B4, C4, D4, E4. So these are like alphabetical order, pretty easy, right? But remember B4, because before we go anywhere else, remember what B4 does, it attracts neutrophils. Before we can do anything else in the inflammatory pathway, we need to get some cells in and some stuff in here that can do some cleanup work. And so we want to, B4 attracts neutrophils. So leukotriene, B4, attracts neutrophils. Now all of these others, so there's some competition going on because these leukotrienes can cause vasoconstriction. We've been trying to vasodilate, but these leukotrienes can cause vasoconstriction, but they also increase vascular permeability. And so even though there's a little bit of competition, there's not too much because we're all still trying to break open those the vessels. We're trying to break open those endothelial vessels on the venule side of the capillary. The really sad thing is, I think this picture is for real. I think someone really made this, don't let it stop you. Which is good, if you say it don't let it stop you, but if you don't let it stop you, it doesn't sound good. Okay, so let's talk about what vasodilation is going to do really quick. So first, we have a blood vessel here, and inside that blood vessel we have some RBCs, and we got some white blood cells, which I'm going to draw blue. And as long as the flow is laminar, and there's nothing going on, all the heavy stuff will stay in the middle, and the edges will be mostly fluid. But as soon as this thing opens up, bam, bam. As soon as that opens up, what's going to happen? Stuff's going to become turbulent. And when it becomes turbulent, what's going to happen? So everything's going to start marginating. All of the white blood cells are going to start being pushed toward the endothelial cells. And then, so that's called margination. We got the first step. That vasodilation is the first step in getting those neutrophils into your tissue. And at that point, if we have all of the right chemokines and cytokines and everything being expressed like it should, we'll get some selectins first, and then we'll get integrins second. So selectins cause things to slow down and start rolling, and then integrins cause it to freeze, freeze, like a stop sign. But I'm colorblind, so it doesn't really matter to me that I picked the wrong color, even though I'm not completely colorblind. I know that's blue. Anyhow, so selectins slow down, and integrins will freeze you and integrate you into the tissue. You'll also want to remember that what the selectins are going to adhere to, so I'll just write selectins, they're going to adhere to are sialylated oligosaccharides. So, I can't even spell that. So, sialate, I think I spelled it wrong. Let me just look at my notes. Hold on. Alright, go number 2. Selectins will adhere to the sialylated, that's why I wasn't spelling it right, sialylated oligosaccharides. And then you remember that the endothelial cells, they'll also start making these integrins. What are the integrins? VCAM, ICAM. And what do they adhere to? So VCAM adheres to VLA4, ICAM to LFA1. And so you get the VCAM, ICAM of the endothelial cells adhering to the VLA4, LFA1 of the leukocytes, and they will freeze it and pull it in. But let's just wait, because all of this stuff is kind of happening at the same time, but it does take a while for those neutrophils to get there. Something happens first. So we don't have the neutrophils in, they're still rolling and stuff. We're going to go look at what else is going on. We're going to pause right there. So now we're at the postcapillary venules, so it's going to be in blue. I got these endothelial cells, in here, and what do those postcapillary venules do? All of a sudden they just start splitting apart. So I can just erase maybe some of this. So they just start splitting apart, and now you've got all this leaky space where different cells and fluids can flow into the tissue. And so let me ask you, what's the first thing that's going to flow in? You're going to have, of course, H2O. You're going to have, yes, all of your proteins that you would normally think of in the blood. But the one that you're not thinking of yet, that you should be, is complement. You're going to get complement in there. Why is complement important? Because complement is another, or let's say that not complement specifically, but activated complement is another chemoattractant for neutrophils. Specifically, C5a. So C5a, we're going to go through like a little background on complement just in case you forgot, so don't give me a second. Alright, so just in case you forgot, here's my tissue damage. I don't know yet whether it's infection. I don't know yet whether it's just a foreign body in there. So let's say if it's infection, what's going to happen? First thing, we're going to have like mannose-binding lectin, and if it's an infection that we've encountered before, we're going to have like an IgG or an IgM, and either one of those things are going to activate complement. So we're going to activate complement. If it's just a foreign body, well, if there's just a foreign body, then it's going to be alternate pathway, because as soon as the complement comes up on there, it'll spontaneously just activate all by itself, and there's nothing on that foreign body to turn it off. So it's going to be an activated complement through the alternative pathway. And you remember that all three of these pathways, they come in and they do one thing. They create C3 convertase. So all these pathways make C3 convertase. And what does C3 convertase do? Well, you got this C3 floating around in the blood, and it gets converted into like a C3b, and a C3a is floating off here in outer space. And that C3b and the C3 convertase, all of this stuff, by our powers combined, we create a C5 convertase. And by that C5 convertase, now we got a C5. What's going to go happen now? We got C5, and C5 floats around here. We got C5a that gets cleaved off. It's floating around in outer space, and then we got C5b that stays here, and then all of a sudden you get C6, C7, C8, C9. All this forms a membrane attack complex, and we puncture this thing, or we opsonize it, one or the other. So if it's just C3 by itself doing its thing, it's opsonization. If it's all the way through the pathway, it's because we're trying to kill something. So it's a membrane attack complex. And now we have these two things floating around, C3a and C5a. Remember, both of these, in case we hadn't already done it through tissue damage, both of these activate mast cells and cause mast cells to degranulate. Remember, I said tissue damage, C3a, C5a, and the cross-linking of IgE. Those three things will activate mast cells. So if we haven't already done that yet, we're activating mast cells. And if we have done that, we got some C5a now going to go out and attract some neutrophils. Now, another paradigm, we could have done this whole thing already. We could have done it with an infection. So a bacteria, we have activation through TLRs, and through the TLRs we create IL-1 and IL-6 and TNF-alpha, and IL-1 and TNF-alpha cause the vascular permeability and the vasodilation, and then they also all go to the hypothalamus. We'll talk about hypothalamus in a minute because I haven't even got to fever yet. So another paradigm would be all the same things would be happening with the bacterial infection. If the infection didn't already cause tissue damage and trauma, activation of arachidonic acid, degranulation of mast cells, if none of that even happened yet, we would still get the same result, because we would have the TLRs. Remember, the TLRs, whenever they're activated, they go through a pathway of NF-kappa B. And NF-kappa B, whenever it is activated, it causes the production of IL-1, IL-6, and TNF-alpha. So NF-kappa B is like a transcription factor, in essence. TLR is how that signal for that transcription factor gets there, and these are the things that are transcribed by those cells, and that's how we're going to get our inflammatory pathway without everything I just said. So now we've got leaky vasculature. So I want to go recover everything I said is going to cause leaky vasculature. So first of all is histamine, IL-1, and TNF, specifically TNF-alpha. Also prostaglandins. So if we have arachidonic acid going through the cyclooxygenase pathway, we get the prostaglandins, also the leukotrienes. So if we have arachidonic acid going through the lipooxygenase pathway, we get the leukotrienes, and all of these things cause vascular permeability. So we are trying to get stuff out of the vasculature into the tissue, and that's how we're doing it. We're breaking open these endothelial cells, and we're putting pressure behind them. We're increasing blood flow. Okay, so I just looked at my timer notes. I'm like at 25, 26 minutes now. I'm going to stop here, and I got to stop here. Like, this photo was used without permission. Oh wait, that's my son. He's a cutie. This is Justice Isaac Burkhead, and he's cute. And so everything else, it's in my notes, guys. I'm gonna upload it to Box here in a minute. I'm like, it's 30 minutes until class start, until session starts. So I'm just gonna start here, and here's my my resources. So this is the stuff that I cited and stuff, and you can look at it.